Studying the evolutionary relatedness between organisms, you are looking at phylogeny. And any phylogenetic tree is going to represent a hypothesis about how the organisms in the tree are related. And the best hypothesis is going to be the one that fits all of the available data. Now, the most basic phylogenetic trees are cladograms, which you can easily construct yourself. It's all about looking at shared derived characteristics, looking at traits that come from a common ancestor. You'll typically have one group of organism that serves as a comparison that is known to have diverged before the lineage that you're looking at. So in this example, that group is the lancelet, which is the out group. Okay, the groups that you are studying are the in group. They're going to be suitable groups to look at that are going to be sharing those traits. Okay, so the lancelet, for example, lacks a backbone. So the presence of a backbone is going to be a trait that separates all of the rest. So a cladogram, you can go sort of like a V up and down or branches left to right. And when you branch, you will mark the shared derived characteristic. So pause for a second. What do you think that's going to look like? Okay, so what that really looks like is this. Okay, the lancelet is the outgroup. It does not have any of the traits in common, whereas the trait that separates everything below is the vertebral column, or the backbone. Okay, hinged jaws separate the next step, and so on. You could have branches that come off in other places. This is just a very simple idea. But the biggest difference between a cladogram and what you would normally think of as a phylogenetic tree is that the branch lengths are equal. The relationships are hypothetical based on shared derived characteristics, and it is not necessarily an indication of time. Is it likely that the lancelet separated off longer ago than the lamprey from the bass? Yes, but I don't have that information on this. I don't have a sense of time or a scale of time. Phylogenetic trees, like the one on the right, will typically be representative of that time characteristic. Okay, evolutionary relationships are going to be represented as these dichotomies, these splits, these branch points, with sister taxa being groups that share an immediate common ancestor. And then you go back and back and back. Everywhere they diverge, that represents common ancestry. And the outgroup in this case would be that basal taxon that you can see in the picture here. So the phylogenetic trees, typically the branches are representative of time and amount of change, and they're going to be more accurate if they're based on genetic information. But they are intended to show patterns of descent, not phenotypic similarity. So the closer they are together on these types of trees, the more closely they are related genetically or evolutionarily. The sequence represents a pattern of descent, and they don't evolve from adjacent taxon. They both evolve from a common ancestor. A phylogenetic tree that is properly set up will include all of the organisms and their most recent common ancestor in a single phylum. So a monophyletic group, that's going to be considered more accurate. Okay, a valid clade, a valid group. When you lack information about some members, you may end up with a paraphyletic or maybe a polyphyletic grouping that are going to be incorporating either missing the common ancestor, not incorporating all of them, a variety of mistakes. When you find out that the relationships are showing up this way phylogenetically, you need to know that your hypothesis has been rejected, and further reconstruction of your phylogenetic tree is going to be necessary. You start trying to compare genetic information, which is considered the best source of information for a phylogenetic tree. It starts out pretty straightforward, but there's going to rapidly be an accumulation of genetic changes and mutations over time. So how can I tell that this final thing evolved from the same source? Well, it's hard with the naked eye, but we can use computer analysis to figure this out. We use a program called BLAST to pull DNA sequences. So you can see an example of that here. The database hits are shown aligned to the query of search, with the aligned sequences the most similar showing as the closest. So there are multiple positive matches here, with those 3 red bars scoring the highest matches. The next 12 represent lower scoring matches, with residues that are falling in that pink range there. And the crosshatch are going to be regions of similarity, but where the intervening area does not match. Okay, so BLAST is going to allow you to study those relationships, looking at that genetic information, maybe from nucleic acid sequence, maybe from amino acid sequence. A variety of structures can be done here, but it's much easier to do with a computer system. Trying to actually compare phylogenetic trees, we typically use a couple of different rules. The first is the rule of maximum parsimony, which says that all other things being equal, the simplest explanation is true. Characteristics are most likely to evolve once. Okay, so parsimony focuses on minimizing the total character states, the total number of changes necessary. So when you're trying to draw the most parsimonious tree, then the shortest tree is going to be the most parsimonious. According to that principle, basically, the simplest explanation should be right. So if you're basing your trait something like on morphological characteristics, the most parsimonious tree is going to be the one that requires the fewest evolutionary events to occur. So looking at the picture here, the one on the left assumes the GT mutation happens once. The one on the right assumes the GT mutation happens twice. So the one on the left is going to be considered more parsimonious. The rule of maximum likelihood says that assuming that mutation rates are equal in the different lineages, most closely related organisms will have the fewest difference in genetic sequences. So maximum likelihood is looking at probability rules about how DNA sequences change over time, and a tree should reflect the most likely sequence. Likelihood methods are complex and they use a lot of information, but if you have a tree that is equally parsimonious, a tree that assumes that DNA changes have occurred at equal rates along the branches is going to be more likely than one that assumes that they are different. So the one on the left is assuming an equal rate of change versus the one on the right, so that would be considered the most likely. Maximum likelihood is one of the most used statistical methods to analyze phylogenetic trees based upon probability models taking mean and variance into account. But that is what you're looking at, rate of evolutionary change. Looking at the example on the left, you have 3 different species with 4 different known nucleic acid sites. Okay, and they have changed over time. There are 3 possible arrangements for this hypothesis in comparison to the ancestral sequence. So this bracketing approach can help you predict things like parsimony. Okay, so if I plot out these changes, okay, well, each of my possible trees, the first nucleic acid is a C, that is a mutation change from the A that the ancestor had. Okay, I plot out the second, the third, and the fourth. And you keep going through all of them, and you're looking at the number of results. So the first tree took 6 evolutionary events to occur, the other 2 took 7. So the first tree would be the one that is the most parsimonious. The biggest problem with parsimony is that it's not always right. Just because something is simple doesn't mean that it is the right answer. People use the I as a point of debate for a lot of this. So because the most parsimonious tree is always the shortest possible tree, that means in comparison to the true tree that actually describes the evolutionary history, the best may underestimate the actual change that has occurred. It's not guaranteed to produce a true tree. They consider the eye too complex to have evolved independently, but we have found that the eye has evolved multiple times in different species in different ways. While eyes can be incredibly similar, like on the example on the left, you have the human eye on the top right and the octopus eye on the bottom left. They have very similar structures, but they are evolutionarily different. They did not evolve from a common ancestor. And one of the biggest differences is going to be that blind spot at the back of the human eye that is not present in the octopus eye. But we find that it gets really super complicated when you're trying to study these trees, trying to study the relationships. Recent research reinforces this concept when you take into the idea of horizontal gene transfer, especially. So we have these trees of life that are sort of this simple branch dichotomous relationship, but unfortunately it's not. Okay, horizontal gene transfer, a 2008 analysis indicated that on average 80% of genes in 181 prokaryotic genomes had moved between species at some point during evolution. So the occurrence of horizontal gene transfers helps explain why universal genes built from different genes give inconsistent results. Basically, we are suggesting that early horizontal gene transfers were so common that early life is more of a tangled web than a dichotomous branch. Some of them have even just given up and just turned the thing into a circle instead. And it's going to take a great deal of information for us to find out the actual relationship. Hundreds of genes have been studied at this point, and we have found examples of fusion and endosymbiosis and horizontal gene transfer. And that complicated of an evolutionary relationship cannot be tricked in a simple tree of life. It's more complicated than that. Life is complicated.